In this section, we're going to talk about G-protein coupled receptors, also known as GPCRs. G-protein coupled receptors are only found in eukaryotes, comprising the largest known class of membrane receptors. Fortunately, humans have more than 1,000 known different types of GPCRs, and every one is specific for the required function. GPCRs can regulate the immune system, growth, our sense of smell, taste, visuals, behaviors, and the mood changes. Talking a little bit about the structure of GPCRs before starting off. The most important characteristic of GPCRs is that they have 7 transmembrane alpha helices. We often also call GPCRs 7 transmembrane receptors, as it is such a unique and interesting structure. Here is the region of GPCR that binds the signal, and this is the region that interacts with the G protein. Now we have our cell membrane, and this is the extracellular side, and this being the intracellular side. Labeling as GPCR. Here the name implies they are coupled with the G proteins. Talking casually about G proteins, it contains 3 different subunits. The first subunit or section of this protein is the alpha subunit. The second we call beta, and the third we call gamma. All 3 of these together are called G proteins. Do you notice that the alpha and gamma subunits are drawn with a little tail-looking thing in our cell membrane, and the reason why is because these 2 subunits, our alpha and gamma, are attached to the cell membrane by what we call as the lipid anchors. They are specialized proteins which can bind GTP, guanosine triphosphate, and GDP, guanosine diphosphate. Guanosine diphosphate here, the protein is inactive and so it binds GDP, guanosine diphosphate. Our first step here is of course that the ligand, which is a green circle, the signaling molecule which binds to our GPCR. Once this ligand binds, our next step is that the GPCR undergoes a conformational change. Showing our 7 alpha helices, it is slightly tougher to draw a conformational change, but the protein is going to look completely different. Now the conformational change has occurred. The protein conformation of a GPCR will alter. Our alpha subunit exchanges GDP for GTP. The molecule is swapped out. Instead of GDP, we have GTP. Now, because we have GTP bound to this alpha subunit, it will now cause our alpha subunit to dissociate and move away from our beta and gamma dimer. Now 2 different sections are formed, our alpha subunit and our beta gamma dimer. These 2 together are going to find a protein in the membrane and alter and regulate the functions of that protein. Both the alpha subunit and the beta gamma dimer can interact with the other proteins to relay messages. Once this alpha subunit activates a target protein, this target protein can then relay a signal. As long as this ligand is bound to the GPCR, alpha subunit dissociates, looks for a protein, and regulates that target protein, causing a whole chain of events that can happen repeatedly. Now, how can we make this thing go back to normal? The next step is that our GTP is hydrolyzed to GDP. Our GTP loses a phosphate in hydrolysis and becomes GDP. Once this happens, everything goes back to normal and the ligand will leave, and everything will go back to looking the way it was and ready to combine with other ligands in the future. Our flight and fight response involves epinephrine and adrenaline, which is the most common example of GPCR. Our GPCR is our adrenergic receptor. Once this epinephrine, a green signaling molecule, binds to our adrenergic receptor, this will undergo a conformational change. It will swap out this GDP on this alpha subunit for GTP. And this alpha subunit will now seek out this other protein and regulate its function. The protein that it seeks out is going to be called adenylate cyclase. What the adenylate cyclase will do is it will take up ATP, adenosine triphosphate, and it will produce cyclic AMP, the cyclic adenosine monophosphate. It will take away two phosphates from our triphosphate and it will make the monophosphate. Once this happens, our cyclic AMP will become a second messenger and will now tell our cells to do other things. For example, if you are competing in a marathon, this will cause adrenaline rush facilitated by these channels. In turn, it will increase our heart rate. We'll also dilate our blood vessels to the skeletal muscles. Remember, fight or flight. We need to start running or fighting. Our muscles are going to have their blood vessels dilated. Finally, all these processes is going to require a lot of energy. So we are going to actually break down glycogen to glucose. And remember, this is our biggest group of cell membrane receptors. It is a pretty complicated process.